What is DNA? DNA stands for deoxyribonucleic acid. DNA will encode genetic information for features that are passed down through inheritance. Adenine, guanine are the two pyrimidines, while cytosine and thymine are the two purines. This relationship follows a predetermined order, which means adenine will pair with thymine, while guanine will pair with cytosine. So the first question for the first part is the structure of DNA is the answer is C, because DNA contains two single strands and it will wind up to form helices. So the second question is DNA is made up of repeating units called C, nucleotides. The third question, the D in DNA stands for B, deoxyribose. The fourth question, the bonds that hold the backbone of each strand of DNA together are called. The answer is D. The fifth question, the information contained in DNA is encoded by the B, nitrogenous bases on the inside of the DNA molecule, which is adenine, thymine, cytosine, and guanine. The sixth question, in which direction is DNA read? The answer is D, 5' to 3'. For question number 7, the answer is B, because A stands for adenine. For the last question, if one strand of DNA is AGCTA, the complementary strand would be D, TGCAT, because A will pair with T and C will pair with G. This is the first question for part 2. By knowing how DNA is made, we make it easier for us to grasp how DNA is replicated, translated, and transcribed. So we will understand how the DNA was synthesized. Without this knowledge, it would not have been feasible to develop technologies like CRISPR-Cas9, conduct stem cell, and also DNA testing services like 2,3andMe. For Linus Pauling model, the nucleotides were on the outside of the triple helix and the phosphate backbone was inside. So below me is the picture of Linus Pauling model. So for the Watson-Crick model, which on my left, the nucleotides were in the center while the phosphate backbone was on the outside of the model. For question number 3, the answer is Erwin Chargaff has discovered the rules of base pairing, which means adenine for thymine, cytosine for guanine. So it was very helpful for Watson and Crick to know how each of nucleotide was present in each DNA region. So for question number 4, what is the main scientific weakness you notice in Watson and Crick's paper? So both the statistical methodology and the overall structure of this paper results are insufficient by established scientific standards. This is because probably scientists from the past year has made some research and there is no p-value. For question number 5, the answer is this thesis make use of DNA capacity to divide into 2 pieces and one will act as a template for self-replication. This is because replication is very important because it serves as a foundation for protein coding. Without protein coding, translation and transcription would be impossible. For question number 6, the first researcher is Francis Crick. Francis Crick collaborated with James Watson to identify the DNA double helix structure. He also demonstrated how a single DNA strand may be utilized to encode a specific amino acid. The next researcher is Rosalind Franklin. She created photographs of DNA using X-ray diffraction. The next one is James Watson. James Watson collaborated with Francis Crick to determine the DNA structure. The last researcher is Maurice Wilkins. He generated a concept for X-ray crystallographic DNA analysis. So this is the last question for part 2. Franklin was never informed that Crick and Watson had used her images since they hardly acknowledged her in their article in Nature. So Watson was very reluctant to acknowledge Franklin's experimental work significant in 1953 development of the double helix. So for conservative replication, it will produce 2 DNA helices, one with old DNA and one with new DNA. So for dispersive replication, it will produce 2 DNA helixes which contain alternative segments of old and new DNA. For semi-conservative replication, it reproduces 2 DNA helixes which one will contain new strand and one will contain old strand. For question number 2, in this slide, it shows the difference between conservative, dispersive and semi-conservative based on the first replications and also the second replication. We can see the different amount of gumdrops and marshmallows in each replication. For the optional exercise, the first question, which of the following statements about the use of gumdrops and marshmallows in this activity is true? The answer is A. For the second question, in this activity, DNA molecules containing only original template DNA are represented using A. Gumdrops and toothpicks only. For the third question, the image shows one round of replication for conservative model. So the answer is A, one molecule that contains 0% gumdrops and 100% marshmallows, while the other one contains 100% gumdrops and 0% marshmallows. The fourth question, the image shows after 2 rounds of replication for the semi-conservative model. So the answer is C. 2 will contain 50% gumdrops and 50% marshmallows, while the other 2 will contain 0% gumdrops and 100% marshmallows. The last question for optional exercise, the image shows the first and the second replication for conservative model. So the answer is A. This is the first question for part 4. The slide shows the theories of DNA replication and the Mendelssohn-Staff experiment. So this is how they know where the DNA containing 100% of 14N or 15N. For question number 2, how can you interpret the data collected after 1 round of division? So the answer is there was equal parts of 14N and 15N. The answer for question 3, the DNA density was evenly at the middle level following 1 replication cycle, which is the conservative replication. So the finding is consistent with the expectations of the semi-conservative replication that all DNA molecules will contain 1 15N level and 1 14N level of DNA. For question number 4, the answer is the DNA would have needed together at the top or bottom of the tube. For question number 5, the answer is after 2 rounds of DNA replication, 2 different regions of DNA were detectable. This is because according to the semi-conservative model, this outcome should have been achieved. So it is very accurate to use the semi-conservative model. For question number 6, what would the data have looked like if the DNA had in fact been replicated according to the model just ruled out? So the answer is only the center contains the DNA strip. So for this question number 7, the answer is only a little strip of mixed 14N and 15N DNA would remain at the bottom of the tube, while the rest would have been less dense than 14N DNA. So this is the last question. Why are bacteria like E. coli good for studying DNA replication? The first point is that E. coli are smaller genomes compared to eukaryotes. The next one, E. coli bacteria contain 4,400 genes, while the humans have 30,000 genes. The third point, that E. coli grow quickly because it reproduces once every 20 minutes.